when I started this project in Avi's lab, my goal was to basically generate a system where I could study the cell biology of neuronal ER. Because at the time, I felt like there wasn't a lot of high resolution images, especially presynaptic terminals. And I think this is really important because uh, uh, proteins that shape the ER, as I'll talk throughout my talk, have been linked to neurodegenerative diseases. However, what are the specific neuronal functions of these proteins is not well known. And so, but before I jump into talking about neurons, I wanted to highlight some of the work that has been done in non-neuronal cells that lays the foundation for the work that I'm going to be telling you about today. And so the ER is this extremely large and highly interconnected network of hollow tubes that's highly dynamic. And here I'm showing a cell expressing an ER marker, and you can see how all these tubules and networks move throughout the cell. And there's three main different types of morphologies that the ER takes. And the one that I'm going to be focusing on primarily is on the tubular ER, which is mostly found toward peripheries of cells, including neurons. And the ER is also a highly social organelle. It has many friends. And towards the end of my talk, I'm going to be specifically focusing on how the ER might regulate neuronal endosomes and some of the findings that we've made there. And so to disrupt this peripheral ER structure, I decided that I wanted to look at mutants of this protein called Atlastin, which is a GTPase that regulates the homotypic fusion of ER tubules and also their tethering. And Atlastin is actually one of the most commonly mutated genes in a type of neurodegenerative disease called HSP. But in non-neuronal cells, when people have looked at elastin null mutants, they've found that these cells have ER networks that are not as interconnected as observed in control cells, where you see this very beautiful honeycomb structure. But surprisingly, when we look at elastin null mutants in neurons, we actually see distinct phenotypes that are separate from this non-neuronal phenotype that I just described. So in the soma in the dendrites of C. elegans, the Sheng lab at Stanford found that the ER at dendrites actually have this enhanced retraction. The Blackstone group at MGH actually found that in mouse cortical axons, there's this transverse ER expansion or kind of ladder-like structure arising in elastin mutants. And then finally, at presynaptic terminals in Drosophila, the Magnu lab at Rice University suggested that these terminals have extensive ER fragmentation. However, I am going to show you today that what I think that this lab was actually looking at was a functional phenotype and not necessarily a structural one. And this is going to be the main focus of my talk today. And so as a model system, I'm going to be using the Drosophila larval neuromuscular junction as a model synapse to study neuronal ER. And I'm going to be focusing primarily at presynaptic terminals. And just for a little neurobiology vocabulary, I'm going to be focusing also specifically at synaptic boutons, which are swellings within this presynaptic terminal that have the machinery that regulate neurotransmission. And so there are many advantages to using this system to study neuronal ER, such as that it's easily accessible for live super resolution imaging, which I'll show you today. It's also about 10 times larger than mammalian central synapses, and this being a glutamatergic synapse is a good model for these. And we can also visualize, you can imagine ER tubules are very thin and small, and so it allows us to really be able to visualize these structures more easily. And then finally, Drosophila elastin null mutants specifically the adults, exhibit HSP-like motor phenotypes, such as difficulty crawling or emerging from their pupa cases, which make this system ideal for potentially identifying phenotypes that could be linked to HSP, as I will tell you today. And so when I started this work, there weren't really any tools to be able to visualize live using super resolution microscopy. So if I can tell you more about this if you're interested, but I figured it out and I was able to basically acquire high resolution movies at presynaptic terminals of Drosophila for the first time. And I want to make a point that I'm going to be actually labeling the ER using two different probes, either a GFP that it's targeted to the ER with BIP chaperone ER targeting sequence, or an ER retention sequence to visualize the lumen. And then to visualize the ER membrane, I'm going to be using a probe of Sec61 beta that's tagged with TD tomato. And so, as I mentioned before, this previous group made this observation that presynaptic terminals of Drosophila Atlastin mutants had this diffuse labeling pattern. But when I started this project, I realized I was thinking that we should also look at the membrane because the ER is this hollow tube, right? And so we could visualize both the membrane and the lumen. And so if ER tubules are highly fragmented, as previously thought, we would expect that both the labeling patterns of the ER lumen and the ER membrane would look similar. But in contrast, if the ER membrane permeability was altered, we would actually see distinct phenotypes or distinct labeling patterns when expressing either a luminal marker or an ER membrane marker in these Atlastin mutants. And that's exactly what we saw. And so I'm going to take you through that data now. And so I labeled the ER membrane using this TD tomato Sec61 beta marker. And on the right, I hope that you can see that these elastinol mutants actually have robust ER networks. We compared to controls. And we actually also looked at to see if there were any changes in dynamics. And actually, to our surprise, found that Atlastin mutant ER, synaptic ER, doesn't really have a lot of defects, which was super surprising to us because as an ER shaping protein, we expected for this to be a much more dramatic effect. And so then using our super resolution system, we looked at the ER lumen, and I was actually able to distinguish two different phenotypes in these Atlastin mutants. So either mutants showed this partial loss of ER networks labeled or complete loss of the networks labeled. And I hope that you can appreciate from these images that the luminal marker seems to be redistributed to the cytosol or a different compartment because it can now diffuse to the periphery of cells. And we were able to quantify these, and we saw that most Atlastin null mutants showed one of these two phenotypes. And we also measured the levels of the marker, and we saw no change in the levels of the marker itself within the synaptic terminals, suggesting that it's just being redistributed to another place that's not the ER. And so what's really cool about this phenotype is that it's specific to presynaptic terminals. We looked at the soma, we looked at the axon, and we even looked at different cell types such as the muscle and found that there was no evidence of this phenotype there, suggesting that this is a presynaptic specific dysfunction. And so up to this point, my hypothesis started to be that maybe the luminal marker is actually being displaced to the cytosol in these Atlastin null mutants. And so if this hypothesis was true, the movement of the luminal marker would be faster in Atlastin mutants because it would be free from diffusing within the ER. And in the cytosol, it will be able to diffuse much faster. And so I decided to use FRAP to test this hypothesis. And I wanted to measure two specific parameters by fitting the recovery of the frapped region to a nonlinear equation that would allow me to measure the mobile fraction of these luminal ER marker proteins diffusing back into the frap region, which would be the plateau. And also looking at the half-life, because that will tell us how fast these proteins are reappearing within the FRAP region. And so when we looked at Atlastin mutants that showed this partial loss of EARC networks labeled, we found that the plateau was reduced and also the half-life was increased, and suggesting that there's less of these molecules diffusing back into the FRAP region, and also they're diffusing much slower. And so one hypothesis for why this is happening is, so if you can see in this inset down here, there's regions that don't reappear in the post-FRAP, part of the movies that I acquired, suggesting that there's both some fragmentation of the ER network in addition to just this diffuse cytosolic redistribution potentially of the marker. And then when we looked at Atlastin mutants that had complete loss of networks labeled, we actually saw that the plateau didn't change. So the mobile fraction of how many luminal ER marker proteins are there. However, they diffused into the FRAP region much faster, again, suggesting that these molecules are now in an environment that allows them to diffuse much faster, which could be the cytosol. However, this is not direct evidence for that. And so I wanted to gain just be able to directly evaluate whether these proteins are really in the cytosol now. And so to do that, I expressed this protein called MorphoTrap, which is a plasma membrane tethered nanobody developed by the Affolter lab at the University of Basel. And the idea was super simple. So in the presence of this MorphoTrap, if the luminal proteins are now in the cytosol in the elastin null mutant, they should bind to MorphoTrap, but not in the wild type where the luminal ER marker is sequestered within the ER membrane. And so to gain an idea on how this binding will affect the dynamics of the luminal protein, we turned again to FRAP and found that the MorphoTrap actually had really low recovery, suggesting that if the luminal marker were actually to bind to this membrane tethered nanobody, it should slow down its diffusion, which is exactly what we saw. So in this bottom graph here, green is the Atlastin mutant, and you can see that both the mobile fraction is reduced and the half-life. And so this provided direct evidence to us that these luminal proteins are actually in the cytosol. And so what is the mechanism of synaptic luminal displacement in Atlastin null mutants? And the answer is we're still working on it. There's a long list that I'm going to take you through now of things that it isn't. So this is very much work in progress, but it's still a very interesting discovery because up to now, we didn't really know that this type of phenomena could occur in neurons because it's previously been shown in cancer cells and in yeast, but not in neurons. And additionally, like I mentioned, elastin and all these other ER shaping proteins are linked to neurodegenerative diseases. So what this phenotype could be, it would be interesting to see if it could be related to disease. And so mechanisms that we've ruled out so far. Apoptosis-induced ER permeability, we don't really see any, or we don't see at all any neuronal death debris at presynaptic terminals, which completely rules out this hypothesis. We don't think it's a defect in translocation because if it was, we would expect to see the same phenotype in the soma, in the axon, and the presynaptic terminal, but we don't. Oops. Oh no. We also tested whether defects in proteasomal degradation could be leading to the accumulation of these proteins in the cytosol. However, we found that we tested this and Atlastin null mutants have no defects, and proteins don't accumulate in controls following inhibition of the proteasome, suggesting they actually accumulated within the ER. So we ruled out this hypothesis. We actually also tried inducing ER stress with DTT to see if that would induce the displacement of luminal proteins in controls, and that also did not lead to the displacement phenotype that we see in Atlastin mutants. So it's not just like an activation or not of ER stress. And finally, we don't think this is canonical ERAD, which is ER-associated degradation, because we still see the proteins in the cytosol in Atlastin null mutants. And we've manipulated the levels of the probe that I'm expressing to visualize the luminal ER. We've looked at earlier larval stages, and we still see the phenotype. So we don't think that it's canonical ERAD. So what I think is happening maybe is there's this type of ER stress that was described by the first Papa group called ER to cytosol signaling, ERSIS or reflux, where luminal proteins through an unknown mechanism get targeted to the cytosol, but these proteins actually retain their function. They're functional proteins that can have new functions in the cytosol. And this was first described in yeast. And so on the left, I'm showing just a yeast cell, just expressing an ER marker, and following tonikamycin treatment, the marker gets redistributed to the cytosol, which is very similar to what I described to you guys here. And so just to end real quickly, we're really interested in understanding how this impacts synaptic function. And so like I mentioned really early in my talk, I'm really interested in understanding how the ER regulates neuronal endosomes, because we know from non-neuronal cells that ER endosome interactions have many different functions. And so I had a really talented undergrad that I supervised, Tina Oken, that she examined all sorts of different compartments and cargos to identify potential, disrupted cargos or compartments within the Atlastin mutant. And what she found was that there's this BMP receptor called thick veins, that its levels are reduced in Atlastin mutants. And this was really interesting to us because previous work has shown that Atlastin mutants actually have increased BMP signaling. And then this led her to also look at the dynamics of this BMP receptor, either in axons or at the NMJ, and surprisingly found that there was no change in the dynamics at the NMJ, but or at the presynaptic terminal, but yes, in the axon. And so this has led us to think that, and it's the hypothesis that we are exploring right now. And I'm definitely going to be using Hannah's probes probably to assess potentially if there are any misrouting defects occurring in Atlastin mutants that either block things from getting to where they're supposed to, or enhancing or creating even new compartments. And so that's kind of where we're right now. And so just to summarize what I've told you today, I discovered that Atlastin null mutants have this synapse-specific displacement of luminal proteins. And, and to our surprise, these animals actually have mild ER network structural defects in aerodynamics. And finally, throughout our work, we're seeing that there's this very striking functional specialization of the ER across either cell bodies, axons, and presynaptic terminals. And there's still many questions to be answered, which I would love to be able to pursue in my lab someday. And so with that, I wanted to thank my lab and my funding sources. And if anyone has questions, let me know.
Thanks a lot, Monica, for that. That's okay. Thanks for the talk. And of course, we're open to questions.
No, why can I?
We're seeing your full. Just that you know.
There we go.
Okay, good. I see there's a question. Yeah, there's a question in the Q&A saying, nice talk. What is the evidence that the luminal protein enters the ER in the first place?
So that's a great question, which I have been asked before. And I mean, other than we're targeting it to the ER, I do have an answer. So that morphotrap experiment that we did, I don't have the slides to show it, but we saw that actually when we are able to pull out basically the cytosolic luminal ER marker, we're able to see ER networks again. So actually, this complete loss of ER networks phenotype is not entirely real because it's just at the resolution of our imaging, we're able to only see just this diffuse labeling. I'm going to go back. I didn't answer that great, but I think that the proteins are being targeted to the ER. It's just that because they're getting redistributed to the cytosol, they kind of mask what's inside what's left basically of the ER. But with that morphotrap experiment, we were actually able to basically recover the ER networks that are in the Atlastin null mutant. Sorry
So
for the
maybe
convoluted. Yeah.
as a follow-up, do you think that the protein that is still in the lumen of the ER exchanges? So there's some kind of dynamic exchange between the lumen and the cytosol of the pool of protein? Or you
Oh my
think that
God.
the one that got out stayed out and will never get be in again? So it's some kind of active extraction. So if
I
you do
guess
some kind of
I don't have I haven't tested that. I mean, we have tried to find endogenous proteins to see if they and we don't really have the appropriate tools right now because since this is an in vivo system, we can't really use antibodies because it will label the muscle ER, which is much more extensive than the neuronal ER. But that's a great question. Yeah, I don't know if there's bidirectional exchange of these molecules. Um, Damn, that's a great question. Yeah, I don't know.
Okay, thanks. There's a question by Patrick Neo Alinaya. Great talk. Have you looked at other ER shaping proteins like reticulins, RIBs, LNPKs, and see a similar phenotype, or do you think this is specific for Atlastin?
Yes, I have looked at reticulin mutants and they do not have this phenotype. So using the same tools, same markers, they do not have this displacement phenotype. So I think this is specific to ala elastin null mutants. And if this is also a neuronal specific phenotype, because if I knock down elastin presynaptically, I still see the phenotype. So there's something special about elastin specific at the synapse that is preventing these luminal proteins from getting out of the ER? Yeah, that's a great question.
Thanks. Next question is from Daniel St. Johnston. So he asks the reverse as my question. So the cytoplasmic proteins get into the ER. So have you seen any proteins that are cytoplasmic now in the lumen of the ER
Hmm.
in the Atlastin knockout?
No, I haven't tested that, but I did think initially when I started this project that maybe what was happening is that maybe there's holes or something in the ER membrane and
Mhm.
things are getting out that way. But I don't think that's what's happening because if you just have like normal flow of things coming in and out of the ER without it being regulated, you would get things like calcium and things like that to the cytosol that would definitely, I think, lead to a dead cell. Right, because the ER
Mhm.
is
Sure.
like the calcium store of the cell. And so I don't think that I think this displacement is active. But I mean we since we the closest thing again that I found that kind of resembles this phenotype is that ER to cytosol signaling or ERSIS um, ER stress response. But we still don't know the machinery. So what I'm planning to do in the future is to, because now I have a genetic handle on this, right? I am able to express an RNAi or look at these mutants and induce this displacement. I want to try to see if I can find either inhibitors or enhancers of this phenotype to kind of start to genetically dissect out what's going on. But as you can see, a lot of my hypotheses haven't been good or no,
Well,
haven't been true or
hopefully you
haven't
can
been
study them in your
validated.
own lab
So
soon. Yeah.
yeah, so definitely this is a little it's more complicated than we thought originally, but it's very, very cool phenotype. So really looking forward to see what we learn in the future.
Great. Thanks a lot, Monica, for the nice talk and
Thank
nice
you.
discussion.